Hello, this is Dr. Morris. I'm doing a media review for microbiology. The course number is MCB 2000.050 and MCB 2000 L.050. This is a review for Exam 1. The chapters that will be covered on the Exam 1 exam include Chapter 1, 2, 3, and Chapter 4. Some of the material I want you to be familiar with for the purposes of the exam will include being able to name and identify the three major domain systems. You should be able to clearly identify those and be able to recognize the description of each of those three domains. There are also a number of scientists that were discussed in the early reading in these chapters. Those scientists include Pasteur, Van Leeuwenhoek, Koch, and Hoek. Also, Virchow was discussed, Lister, as a number of scientists that were also discussed in the early reading of those chapters. You should be able to appreciate the contributions of each of those scientists. I'll go over some specific examples as I go through the review. First, Louis Pasteur had a significant experiment where he demonstrated the use of what's referred to as the S-neck flask. Please be able to describe that flask and be able to describe the importance in its contribution to microbiology. Also, Pasteur was very important in disproving the theory of spontaneous generation. You should be able to identify for me the theory of spontaneous generation, what that theory proposed, and then how Louis Pasteur was able to disprove that theory. You should be able to appreciate and describe the contributions of the scientist Hoek, H-O-O-K-E. You should be able to describe the contributions of Van Leeuwenhoek, specifically what his contributions contributed to microbiology and the study of microorganisms. You should also be able to give me a comparison and contrast between the organisms in the category of viruses and bacteria. There are some notable differences between viruses and bacteria, and so you should be able to compare and contrast those two organisms. You should also be able to give me some contributions of the scientist Jenner. Jenner was very important in identifying a specific process that we use even today. He was able to develop this technique after identifying some specific characteristics of microorganisms. So you should be able to explain the contributions of Jenner and his contribution in the field of microbiology. You should be able to define the term normal microbiota. Normal microbiota. This term also is commonly referred to as normal flora. You should be able to describe and identify what is meant by that term. You should also be able to compare and contrast the following microorganisms. You should be able to give me the general characteristic of algae in comparison to bacteria, general characteristics of fungus in comparison to bacteria and algae, helminths and their characteristics in comparison to the other microorganisms, and as I've already noted, viruses as well in their comparison to the other microorganisms. You should be able to identify what is meant by the term specific epithet. Specific epithet. And as you know, many of the microorganisms are referred to in their specific epithet. And what is meant by that? You should be able to identify the difference between bacteria and archaea. There is a specific characteristic difference between archaea and bacteria, and you should be able to identify that. You should be able to describe some of the chemical contributions that we see in the study of microorganisms. This includes a number of various bonds. This includes ionic bond, covalent bond, and hydrogen bonds. You should be able to tell me the difference between those types of bonds. You should be able to give me an example of an ionic bond. You should also be able to describe the chemical structure of carbohydrates. You should be able to describe the chemical structure of proteins, chemical structure of lipids, and the chemical structure of nucleic acids. You should be able to identify a hydrogen bond, and you should be able to identify what's meant by the term covalent bond. I've already asked earlier about you being able to describe an ionic bond. You should also be able to give me the general characteristics of nucleic acids. I may have said that earlier, so if I'm being redundant, I apologize. You should be able to describe what is meant by the term polymers. Give me an example of polymers. Give me an example of polymers related to glucose. You should be able to describe the characteristics of the organism or the bacteria, Desulfovibrio. Be able to give me the general characteristics of this bacteria, what type of environment it lives in, and some specific characteristics. You should also be able to give me a description, and this relates to current events, of oil-degrading bacteria. Now, these bacteria are naturally present in the environment, but there are some specific reasons why these microorganisms literally cannot degrade significant amounts of oil in a short period of time. But there are some mechanisms in place that might help these microorganisms speed up the process, and you should be able to describe this. And this is in your reading. You should also be able to describe to me the gram stain. You should be able to identify the steps of the gram stain, and if asked, should be able to write out each of the steps of the gram stain in the correct order. Now, in the gram stain, several chemical products are used in the process. You should understand the importance of these chemicals that are used in the process of gram stain. Now, you should also be able to understand the results of the gram stain. In the gram stain, organisms can be identified based upon a gram-positive result or gram-negative result. You should be able to define that for me, describe it. You should also be able to tell me the characteristics and the appearance of a gram-positive and the appearance of a gram-negative microorganism. Also in your reading is a discussion about various types of microscopes. The compound light microscope, the phase contrast microscope, the dark field microscope, the fluorescent microscope, and the electron microscope. Each of these microscopes have specific characteristics. They have specific capabilities and are able to view specific microorganisms or the parts of specific microorganisms in a specific way based upon the type of imaging that is set up for these microscopes. So you should be able to clearly describe the use of this equipment or these microscopes and how they are used to identify specific microorganisms. There are also some other aspects of microorganisms that are important as it relates to staining. Now, we discussed the gram stain, but there are also other staining mechanisms available as well, one of which is referred to as the malachite green stain. You should be able to identify that process and what specific microorganisms or what specific characteristics of a microorganism does this help to identify. In the process of observing microorganisms under a microscope, a bacteria smear must be performed. You should be able to explain to me what is meant by a bacterial smear, and the steps are involved in that process and why it's so important. As we talk about the light microscope, you'll be asked in your lab to identify the various parts of the light microscope and the importance of these parts, what the function of the various parts of a microorganism are. You should be able to identify the purpose of the ocular lens, the purpose of the stage, contrast, the importance of a refractory index. You should be able to identify the importance of each of the parts of that microscope. There are some other terms that are also important in the study of microorganisms that you should be familiar with for the purposes of the exam and for our future studies as well. These terms refer to the concentration of a particular solution. The terms are hypertonic and hypotonic. Certain organisms can exist in hypertonic solutions, others in hypotonic solutions. Should be able to tell me the difference. Should also be able to describe to me what happens to particular cells when placed in a hypertonic solution or when they're placed in a hypotonic solution. You should be able to define osmotic pressure. Again, the definition for osmotic pressure. Now, not only should you be able to recognize that definition, but you should be able to understand the importance of osmotic pressure in bacterial life. Also, you should be able to describe mechanisms of how substances are transported from areas of lower concentration to higher concentration. Some of the terms that you should be familiar with and be able to identify include simple diffusion, facilitated diffusion, active transport. You should be able to also describe to me the general characteristics of the plasma membrane, the role of the plasma membrane or the function of the plasma membrane, and exactly what the constituents are and the importance of the plasma membrane. You should be able to describe to me the general characteristics of the endospore, why the endospore is so important in certain microorganisms, and we discussed earlier some of the techniques involved in how to identify organisms with endospores as well. You should be able to identify the bacterial sensitivity of certain microorganisms, and I'll lump them in categories. Gram-negative organisms respond to certain antimicrobials, and gram-positive organisms respond to certain gram-positive organisms respond to certain antibacterial therapy as well. You should understand the general responsiveness of both gram-negative and gram-positive organisms to certain antibiotic therapy. You should be able to describe to me the structure of the glycocalyx. You should also be able to describe to me the bacterial cell wall. What are the components of the bacterial cell wall? Let's see. You should also be able to differentiate or compare and contrast a process of simple diffusion with facilitated diffusion. You should also be able to describe some particular structures noted in microorganisms. These include the fimbria and the pili. You should be able to compare and contrast those two structures. You should be able to compare and contrast eukaryotic cells and their general characteristics of eukaryotic cells. I'm sorry I said compare and contrast, but you should be able to identify and describe eukaryotic cells, their general characteristics. Now, that ends the material that's going to be covered in the lecture portion of the exam. Again, all of those questions are based upon your reading assignments, and the reading assignments cover Chapter 1 through 4. And then that material is taken directly from the lecture slides that are on the course document under the lecture portion of the course. Now, I'm going to move to the lab portion of the course and some material that's important for you to be able to know for the purposes of the lab. As I said during the lecture portion of the review, I will be asking you to identify specific structures on the light microscope. These structures may include the objective lens, corrective lens, the ocular lens, the condenser, the stage. All of those are particular parts of the microscope that I would I would expect you to be able to identify the roles and the importance of each of those structures. I would also expect you to be able to identify gram-negative versus gram-positive gram stains. I would also expect you to be able to identify the process of acid-fast staining. What microorganisms are typically observed after performing acid-fast staining. There is a specific process of acid-fast staining that helps to identify these microorganisms, and they have a specific characteristic that I would expect you to be able to identify. There are specific organisms that use acid-fast staining, and I would expect you to know what those microorganisms are. You should be able to identify the morphology of various microorganisms. And as I describe the morphology, I mean bacillus, cocci, streptococci. All of those are specific morphologies. There are more, and I would expect you to be able to identify those. You should be able to identify the malachite stain. Malachite stain is important for a specific covering on microorganisms. You should recognize what that is. You should be able to recognize that stain. You should be able to identify images related to the various types of microscopy. These include dark field, phase contrast, light microscope, and electron microscope. You should be able to identify images that are related to each of those types of microscopy. I would expect you to be able to give me the general characteristics of some of the microorganisms. These include Mycobacterium tuberculosis, also includes Escherichia coli. These have specific staining properties that you should be able to identify, and they also should be recognizable. And these are in your reading assignments associated with the lab. I would also expect you to be able to describe some of the general characteristics of microorganisms under electron microscopy. Again, remember, electron microscopy gives you a very high magnification of certain microorganisms, and certain characteristics present with organisms that are viewed in this technique. I would expect you to recognize Neisseria species of microorganisms. They have a specific morphology and also have a specific stain. I would expect you to be able to recognize Staphylococcus. It has a specific morphology and also has a specific stain. I would also be able to expect for you to identify Clostridium. It has a specific stain and a specific morphology as well. This is all the information that is covered in the laboratory portion of your course. Again, these questions are taken directly from your lab assignment and your reading assignments as well in the chapters that I've described earlier. This ends the review for the lab portion of microbiology, MCB 2000L.050 for the first exam. And this whole media file covers the information that is for the lecture portion and the lab portion of MCB 2000, the first exam, which covers chapters 1 through 4. Thank you very much, and good luck in your studying.